Arun Sharma, stem cell and heart biologist at Cedars-Sinai Medical Center's Regenerative Medicine Institute. Arun will tell us about using human pluripotent stem cells to model the effects of spaceflight on the heart, a paper that was published late last year and was conducted on orbit just about 4 years ago this month. Arun.
Well, thank you for that kind introduction, Liz, and thanks to the National Lab for giving me the opportunity to talk a little bit about our work that we've done over the last few years, utilizing human pluripotent stem cells to model the effects of spaceflight on the heart. And I'll be recapping some of the work that I conducted as a graduate student at Stanford University in the Cardiovascular Institute, but I'll also briefly touch on some of the ongoing work that we're doing here at Cedars-Sinai. So the cardiovascular effects of spaceflight, why are we actually interested in doing this work? Well, we have an understanding as to what happens to the heart as a whole, as a tissue, during extended microgravity. There's an understanding that there's a reduction in overall cardiac work, as well as an increase in heart muscle loss. And this is as a result of that reduced cardiac work. And also as a result from that muscle loss, there's a shape change that actually occurs in the heart. The heart actually assumes more of a spherical shape on orbit as opposed to the traditionally fist-shaped structure of the heart. So we have an understanding of what happens to the heart at the organ level, but we have less of an understanding of what actually happens to the heart at the cellular level. And so we are using a couple of revolutionary pieces of technology in stem cell biology to really help address some of these issues and help us better understand what happens to the body at the cellular level in microgravity. Okay, so we are using induced pluripotent stem cells, and for those of you who are on this call and have an interest in stem cell biology, these cells are of course no surprise and nothing new to you, but just as a quick recap, induced pluripotent stem cells are a cell type that is largely equivalent to the embryonic stem cell. These are cells that are able to be produced from a small sample of an individual's own skin or blood. They can be reprogrammed to induce pluripotent stem cell colonies, and then from there differentiated into a variety of different cell types, such as cardiomyocytes, the muscular cells of the heart. And of course, the fun thing to note about these cells is that since they are cardiomyocytes, contracting heart muscle cells, they're able to spontaneously contract in a dish. There are a number of different applications for these cells, including potentially for autologous cell therapy. But the focus of this presentation and the focus of most of my work has been on using these cells for disease modeling, as well as for drug screening and drug discovery, and using these cells for examining the effects of microgravity as an analog for the human heart. So I'll give a brief overview of some of the work that we did over the last few years. We had the good fortune to receive some funding from the Center for the Advancement of Science and Space and CASIS to derive iPSCs, induced pluripotent stem cells, from 3 separate individuals, and then differentiate those cells into contracting cardiomyocytes, then transport those cells to the International Space Station via a SpaceX Falcon 9 rocket. Of course, we also had a parallel set of ground-side control samples here on Earth so that we would be able to compare the effect of microgravity on our iPS cardiomyocytes on space to a parallel set of conditions. Once we were on orbit, we conducted a number of analyses, including gene expression analyses, looking at how these cells change in terms of their gene expression, as well as functional analyses for contractility, calcium imaging, and structural cellular analyses using immunofluorescence. We also returned a sample of these cells back live to the planet for further analyses, and these results were actually recently published in stem cell reports at the end of last year, so feel free to take a look. I'll give a brief overview of our approach to actually culturing these cells, and also I'll talk a little bit about some of the data that we got from our experiment. This was a very collaborative effort, and our implementation partner in Bioserve Space Technologies helped us develop a custom set of cell culture growth plates. These are fully enclosed 6-well plates that have individual wells isolated to prevent cross-contamination. They're fully enclosed, of course, because we want to avoid media and nutrients and cells from escaping aboard the microgravity environment of the International Space Station. So these were the culture systems that we actually used aboard the International Space Station. Perhaps the greatest assistance and the greatest help that we had for this entire project was from Dr. Kate Rubins, who is not only a NASA astronaut, but also a very talented cell biologist, as Jana alluded to in her talk. We had the very good fortune of working with Dr. Rubins over the course of a month in the summer of 2016. So that we could actually examine the effect of microgravity on these iPS cardiomyocytes. And what Dr. Rubins was able to do was to conduct weekly nutrient exchanges on these cells to maintain their viability, also conduct microscopy, and maintain the iPS cardiomyocytes for 30 days aboard the space station. So what do we find? I'll walk through some of the data and some of the results that we actually obtained from these experiments. Well, first we found at the macro level, the iPS cardiomyocytes did not exhibit significant overt changes in cell morphology or sarcomere structure after return from spaceflight. Sarcomeres are the individual contractile components of cardiomyocytes that actually enable them to contract at the cellular level. As you can see here in this video, the iPS cardiomyocytes did continue to contract during prolonged exposure to microgravity, although there were some changes in the way the cells actually contracted on orbit. So this was the first demonstration of long-term cell culture aboard the International Space Station. Again, as I mentioned, we cultured these cells for 30 days. And one other thing to note is that these are relatively advanced cells. As you can see here, they're functionally active, they're contracting, they're electrophysiologically active cells. So another important aspect of this project was to demonstrate that long-term cell culture is indeed feasible aboard the International Space Station. As I mentioned, we observed some changes in the way the cells actually contracted on orbit. There was a trend downward in terms of their overall beat rate after up to 2.5 weeks on flight. There's also a trend downward in their contraction and relaxation velocities as well. And so this may be a result of the cells adapting somewhat to their microgravity environment. These are functional cells, as you can obviously see through the contraction of the cells. And to elicit the contraction, cardiomyocytes have to undergo an excitation contraction coupling process, whereby the calcium ions within the cells are actually activated, and ultimately these ions are what lead to the individual contractions of the cardiomyocyte. And because of that reason, you can track calcium flow and calcium flux in individual cardiomyocytes. So we did calcium imaging on these space flow and heart cells after they returned back to the planet as another functional indication of how the cells are behaving. And what we did observe is that these cells exhibit reduced calcium cycling after return from spaceflight. Finally, we're able to preserve a sample of iPS cardiomyocytes at multiple time points during spaceflight for RNA sequencing. So this is conducting gene expression analysis of space-flown samples and compare them to ground-side controls as well as post-flight samples, samples that were preserved after the cells returned back to the planet. And what you can see is that the samples that were returned to the planet in the post-flight set of samples here largely show a similar gene expression profile to the ground-side controls, whereas the flight samples exhibit a slightly different gene expression profile based on the cells that are activated, based on the genes that are activated in these cells. And if we took a closer look at some of the gene pathways that are actually altered in those space-flown samples, we identified a number of genes related to mitochondrial and metabolic function. So to conclude, we were able to demonstrate that human iPS cardiomyocytes, human heart cells, could be successfully cultured long-term aboard the International Space Station. As I mentioned, this was the first demonstration of long-term cell culture aboard the International Space Station, and also culture of a complex cell type, a functionally active, electrophysiologically active cell type in human cardiomyocytes. These cells exhibited some alterations in contractility within weeks of spaceflight, although it did not change their overall structure as shown by immunofluorescence. They also showed gene expression changes, and in particular, changes in pathways related to mitochondrial and metabolic function. And ultimately, we believe that this is a useful model system that could be used to model the effects of spaceflight and microgravity on the human heart at the cellular level. So I'll leave you with some future directions, projects that are in the works, and some food for thought for additional analyses that will need to be done. How can we improve this particular experiment and this particular model? Well, this was a two-dimensional cell culture system. Everything that I showed you, all the data that I showed you, was from two-dimensional cardiomyocytes grown in a monolayer. Ultimately, the human heart has a variety of different cell types that are important to its function, not only the contracting heart muscle cells, but also endothelial cells, the support fibroblast cells, and so on. And a better model system, a better multi-lineage model system would be able to leverage these other cell types as well. There are a variety of different improved model systems that are being developed right now utilizing, for example, tissue engineering approaches that can artificially print multiple cell types into a single system. Organ chip approaches that Jana talked about briefly, where you can grow multiple cell types on an integrated organ chip which is connected through a pseudovasculature, and also organoids, these three-dimensional collections of stem cell derived tissues, would also be a useful model system to better understand the effect of microgravity on the human heart. Additionally, there's a need for an automation of processes. Again, as Jana alluded to in her talk, crew time is very precious on the International Space Station. And although we were very fortunate to have an astronaut working with us, and Dr. Kate Rubins, who has a background in cell biology, that's not always the case. Of course, the astronauts have a variety of other tasks to attend to while they're on the International Space Station. They have a variety of different experiments that they're doing for other projects, and also maintenance of the station itself. So if we can further facilitate the automation of some of these cell culture processes for maintaining these cells on orbit, that would be advantageous not only for the astronauts, but also for the scientists as well. And finally, scalability and in-orbit production. Is this something that would be feasible? There is some interest in potentially utilizing the station in microgravity environments as a means to facilitate the production of stem cell populations and stem cell derivatives. This is something that you would have to examine in the context of throughput as well. Can we manufacture these cells at a GMP or good manufacturing practices level so that we could biomanufacture these cells and potentially use them for translational purposes back on the planet. And we've actually had the very good fortune to obtain a recent grant from NASA to further investigate this. This is with our team here at Cedars-Sinai in collaboration with Space Tango, who has been developing a number of these automated platforms for facilitating cell culture on station. And so we're in the process of commencing that particular project. And with that, I'd like to thank everyone who has been instrumental in helping to get these projects off the ground, no pun intended. Of course, collaborators at the Stanford Cardiovascular Institute for the two-dimensional iPS cardiomyocyte projects, as well as collaborators here at Cedars-Sinai and Space Tango, our implementation partners in BioServe, SpaceX, and of course the Center for the Advancement of Science in Space, which has helped fund this particular work. And for additional information on our iPS cardiomyocyte project, our two-dimensional iPS cardiomyocyte project, I would refer you to our recent publication in Stem Cell Reports. So thank you again for your time.